Let's take a look at this exam question which tricked most candidates who attempted it. This question presents us with a multi-step context which we're going to have to interpret in order to answer the following question. State the base sequence on the anticodon of a tRNA molecule that brings valine to the ribosome. Now the question is set in the context of sickle cell disease, in which the hemoglobin which contains the amino acid valine is causing sickle cell, when typically it would be occupied by glutamic acid. Now we may not know anything about those amino acids or their positions in the structure of hemoglobin, normally or otherwise, but we can see in the diagram above that glutamic acid's position as well as its mRNA triplet code has been given to us. The triplet code GAG, coding for glutamic acid, allows us to understand and interpret this genetic coding chart if we have not seen one before. We can see here that GA and then G or A for the third letter in the triplet code provides us with the code for glutamic acid. And so we can understand from this example how this is meant to be read. This allows us to attempt to answer the question, how do we end up with valine instead? Well, valine, we can see, is coded for by G, U, and then either G or A or C or U. And it would be at this point which many candidates would give one of those four possibilities. However, that is the first trap that this question has laid. The question has stated that sickle cell disease is a genetic disease that results from a substitution mutation in one of the genes that code for hemoglobin. This is important information that is often overlooked because it means that only one base has changed from an existing sequence to produce this mutation, which happens to be the amino acid valine instead of glutamic acid. The consequence of this is that to go from glutamic acid to valine, only one letter in the triplet code can change. Now we can get both by starting with G, but in order to get valine from glutamic acid, the second letter has to change from A to U, using up our substitution mutation. Now the third letter, which is coding for glutamic acid in our mRNA strand in figure 16.2, is G and not A. But we cannot change the third letter because we've already used up our one change from the substitution mutation, eliminating the possibility of coding for valine with A, C or U. Therefore, the only possible triplet code which can code for valine is going to be G, U and G. This would seem like the logical answer to give, and again this is where the second trap that this question has laid presents itself, because the question has asked us to state the base sequence of the anticodon of a tRNA molecule that brings valine to the ribosome. Because the anticodon is complementary to the codon on the mRNA, it means that it is not going to be GUG, but is instead going to be CAC. A very challenging question for 1 mark. This tells us that if we are presented with multiple sets of information for a seemingly simple question, we should make sure that we identify all relevant biological mechanisms within it before settling on our answer.